My name is Kristina Sin. I currently work at Dr. Gonzalez's lab at the U of A Phoenix campus, and my presentation today will be on targeting the cerebral vasculature in treating acute ischemic stroke. So stroke can happen to any of us at any time and any age, and it can cause irreversible damage to the brain and even potentially death. And it can happen in two ways. So the first way and the most common way is through ischemic stroke, which is the blockage or the blood clotting of a blood vessel supplying blood to the brain. And to better understand this, we can envision the blood vessels in our brain as railroad tracks that are made of structural units that can be likened to our endothelial cells that line the vasculature. And these railroad tracks carry life-sustaining supplies, that being oxygen and glucose, to the local villages or our resident brain cells. And so when there's a boulder or clot that blocks that supply chain, that village is then going to struggle to survive and slowly starve off and die until that blockage is removed. And then a hemorrhagic stroke or brain bleed is when a weakening of happenings of the blood vessel, and that causes a rupture of it, that allows for blood to leak into the brain and then allows for the compression of tissue and additional damage with that added pressure of that blood accumulating over time. And this is somewhat similar to a faulty railroad track in which the train is going to derail and the supplies are going to spill out everywhere. And so not only is the village going to starve, but it's also going to incur additional damages with that crash, and oftentimes going to affect the village for the worst. And so for treatment options, unfortunately for hemorrhagic stroke, there are only preventative measures available to it that have to be implemented well before the initial ischemic attack or the stroke attack. And then fortunately for acute ischemic stroke, there are two FDA-approved therapies, one being the mechanical removal of the clot via thrombolectomy devices, or the treatment by recombinant tissue plasma membrane activator, or RTPA, that basically dissolves the blood clot. And one thing about these treatment options is that they have a very high hemorrhagic risk with them, and that comes with the conditions of the ischemic stroke in which the blood vessels actually weaken over time as the injury persists. And so oftentimes when stroke patients do arrive to the hospital, they don't even qualify for treatment at all. And so with the initial injury of ischemic stroke, we have the arterial occlusion that immediately induces this hypoxic and glucose-deprived environment that induces then the production of ROS, or reactive oxygen species, to be produced, and the coagulation cascade to take its course. And so all in all, these conditions actually induce neuronal cell death, and then microglia activation, and then the production of pro-inflammatory cytokines like these shown here. And an immediate immune response that occurs is the recruitment of immune cells to the injury site, and specifically neutrophil cells immediately, which are able to adhere to the endothelium that line the vasculature. And so these neutrophil cells actually release granules of this zymogen matrix metalloprotease 9, or pro-MMP9 here. And once that enzyme is able to activate to the MMP9 form, that enzyme is actually able to cleave components of the blood-brain barrier, such as the tight junctions and the extracellular matrix that stabilize that barrier. And these MMP9 enzymes also are able to stimulate more neutrophil recruitment to the site. And so this causes a positive feedback loop in which there's increased pro-MMP9 presence and thereby MMP9 activity at that layer. And additionally, with the pro-inflammatory cytokines that are produced, that actually also increases the activation of endothelial cells that actually increase the adhesion molecule expression, and that adds to the increased neutrophil presence. And so with both the increased presence of neutrophils and increased activity of MP9 that work to decrease the permeability or increase the permeability of the blood-brain barrier, these neutrophil cells actually are then able to transmigrate through the weakened barrier and into the brain. And so in that cerebral space now, we have the vicious cycle of increased MP9 activity to then also contribute to the loss of the blood-brain barrier integrity. And so collectively, this increases the development of cerebral edema, weakens the vascular integrity, and increases the risk of that ischemic stroke to now transform to a much more deadlier hemorrhagic stroke, and overall increasing stroke severity So in all, MMP9 has been strongly implicated in mediating the blood-brain barrier breakdown, which is why my lab is particularly interested in studying MMP9 as a therapeutic target, uniquely at the endothelium layer. So a lot of studies in the past have actually focused primarily on protecting neurons as a therapeutic target, rather than at that protective layer, at that protective endothelial layer that acts as a first defense in this decisive breakdown. And so with that, my work for the summer has been evaluating the inhibition of MMP9 as a novel therapeutic target, an option for reducing BBB integrity loss, and ultimately towards improving stroke patient outcomes.